research project, we optimized genetic algorithm parameters for atmospheric carbon monoxide modeling. I worked under Dr. Hammerling and William Daniels, who is a graduate student also working under Dr. Hammerling. Dr. Buchholz also helped with this project, and she's affiliated with NCAR. The motivation from this project stems from the fact that in the Southern Hemisphere, a main source of atmospheric carbon monoxide variability is large burn events. Therefore, carbon monoxide can be used as a proxy for fires. Multilinear regression models that predict atmospheric carbon monoxide concentrations use something called climate indices. Climate indices affect the amount and dryness of available fuel, which in turn affects fires and hence carbon monoxide variability. So here's the multilinear regression model that was created, where we can see the corresponding variables underneath it. The R package that we created, called Red Climate Chem, has 3 different variable selection algorithms: the exhaustive method, which finds the best model but has a long runtime, stepwise, and the genetic algorithm. So here we look to optimize different genetic algorithm parameters to decrease the runtime and still have a good model accuracy. So the genetic algorithm is a stochastic variable selection technique, and here we can see the general flow of it with the 3 different model modification techniques, and at the end there's a stopping criterion which must be met in order for it to finish. So in the genetic algorithm, we chose these 5 different parameters to optimize, and here we can see the default values and the values studied. We studied these selected parameters with a simple grid search over them. So for example, we started with a 4 covariant model, and here we can see just an example plot of what we did with the population size. So on the left-hand side, this is the proportion of differences, which is essentially the number of models when run with a specific population size that differ from the exhaustive method over the total number of models. And on the right-hand side, we can see the runtime, which corresponds to how long it took to run that run with the specific population size. And so in this case, we would look to both decrease the runtime and the proportion of differences. So we chose 40 as our optimal value. So then we moved on to a 5 covariate model where we did pairwise interactions on a HPC system. So here in this table, we can see our optimized results with the parameters on the left-hand side and the optimized values for the 4 covariate and 5 covariate models. So here is a box plot of the default parameters versus the optimized parameters for the 4 covariate case. And using all optimized values, the runtime decreased an average of 11.8%, and there's practically no change in model accuracy, meaning that the optimized parameters result in a minor decrease in model accuracy, which is really good. So for the 5 covariate case, it was found that the exhaustive method was faster than the optimized genetic algorithm. And so we concluded that the genetic algorithm trends toward the exhaustive search on an HPC system, essentially because the optimized population size that was chosen was very close to that of the total number of models. So that is an overview of our poster, and thank you for listening.